Hello everyone and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. We're in the middle of 3 talks on the theme of bias sampling in phylogenetics. What does it do to our inferences and what can we do about it? We started with a theoretical perspective from probabilist Leah Popovic. Today's talk is from Vladimir Minin concerning a new model that explicitly accounts for preferential sampling. Next talk will be from Nicola Mayo, who will discuss the sensitivity of discrete trait methods to sampling strategy. As I mentioned, today we have a talk from Vladimir Minin. After his undergraduate education in Ukraine, Vladimir did a master's in math at the University of Idaho, followed by a master's and a PhD in biomath from UCLA, advised by Mark Suchard. Since 2007, he has been on the faculty of the University of Washington Department of Statistics and was promoted in 2013. Vladimir works on quite a range of broad statistical topics, including but definitely not limited to phylogenetics. Today he's going to tell us about the impact of bias sampling on changes in effective population sizes and what we can do about it. Welcome, Vladimir, and I'm looking forward to your talk.
Thank you, Eric, for inviting me to give this talk. As Eric mentioned, I'm going to be speaking about preferential sampling when trying to estimate changes in effective population size. This is joint work with a PhD student from University of Washington Statistics Department, Mike Karcher. That's primarily his work. Also, tremendous help was given by Julia Palacios, who is now a postdoc at Harvard and Brown and used to be a PhD student here in statistics. Another 2 collaborators are Trevor Bedford from Hutchinson Center and Mark Suchard from UCLA. And we're grateful for NIH funding these projects. All right, so let me start by just motivating this work. I'm not going to be talking about this next flu project, which is not by any means have nothing to do with it really, but it's Trevor's baby and Richard Nayer's baby. But I would like to just show this slide to show that where we want this to go eventually, all of us, I mean, as a field, we would like to be able to predict evolution, for example, of infectious disease agents in order to take some action. In the case of flu, maybe we would like to develop better vaccines, understanding how the current how the influenza sequences are evolving during the current flu season in order to understand how to deal with them when the next season comes. I argue that in order to predict the future, it's probably good to start by trying to understand the past and the present dynamics of the flu, as if we take flu as an example. And I'm just showing you on the bottom a figure from a important paper by Andrew Rambo and co-authors when they used these coalescent-based approaches that I will be introducing in this talk to estimate sort of population dynamics of flu in different geographic locations and looking at different strains of flu. And on the top of that figure, it's not, so the bottom figure is from Andrew Rambo's paper. The top figure is just kind of superimposed loosely a possible, it's not the same genealogy that was used in this work, although it's very close to it, showing the genealogy relating samples of these flu sequences taken over time. So that's kind of big picture that these what these methods do. They take the genealogy, or better, sample over genealogies, but you can imagine just one simplistic approach would be estimate the genealogy of your sample of sequences, and using somehow the shape of the genealogy, how these coalescent events distribute across time, you would like to infer the changes in the population size. And it turns out it's possible under certain conditions. So in order to understand how it's done, let me briefly introduce the coalescent. The one point that I would like to make is that coalescent is not really a model. Coalescent is a consequence of, or a limit of, a Wright-Fisher model in a certain sense. So it's hard to talk about coalescent without defining the population genetics forward model. You can start with Wright-Fisher or Moran model, doesn't matter. So in the Wright-Fisher model, we just assume a constant population size. non-overlapping generations, and we're imagining just doing sampling with replacement in order to produce offspring of individuals from a previous generation. So in this figure, rows represent generations, and number of circles is your sample size, population size, sorry, or twice your population size, and in some notation. And then each time you have a line between two subsequent generations, that means that that individual produced an offspring that carried on to the subsequent generation. It's a very simple model. We assume no geographical structure, no recombination, no selection, well-mixed population, all of that. We will relax, of course, the constant population size because we would like to estimate it. But and all of these conditions could be relaxed, although relaxing some of them leads to severe problems with inference. For example, relaxing the no selection assumption is still a big problem, and even recombination is computationally very difficult to relax, assuming recombination, when you move to the coalescent. So coalescent, as I said, is kind of a Wright-Fisher model, but run backwards. You start with Wright-Fisher or something like Wright-Fisher, and then you ask yourself, what if you take a random sample from the current generation of your population, which in this figure are marked by red circles, and you ask, what's the can you trace back the ancestry of your sample? And so using the properties of Wright-Fisher, you can sort of derive quickly that two genes coalesce in one generation with probability 1/2n. And so that gives you some probabilistic model of how to coalesce pairs of individuals or pairs of genes. And you can derive that the time until coalescence of two genes in J generations, that's probability will be sort of just geometric looking probability. So the waiting time is geometric, which eventually leads to this Kingman's coalescent approximation. But again, we can interpret the coalescent only because we know that it came from the Wright-Fisher or Moran model of forward model. So you could play some tricks, and you can rescale time to be continuous, in which case geometric distribution becomes exponential. In the case of constant population size, the waiting times until the next coalescent event, when two lineages merge together, are independent, exponentially distributed with the rate equal to k choose 2, the number of pairs that you can select. When you have a non-constant population size, as I said, that's something we're interested in this project. Things become trickier. Your coalescent times are not independent anymore, and they follow something more complicated, and not an exponential distribution. But the point I want to drive is that if you there's a model that you can simulate from that model. You can study properties of that model, which was done by Griffith and Tavare in the 90s. And if you look at the simulation, so here I show 5 randomly generated genealogies with 10 samples with the constant population size versus exponential growth, which is denoted by minus sign as a decline, but it's really exponential growth. You can see that the genealogies have different properties just visually. So for example, you can notice that the red genealogies under exponential growth have these sort of tall legs, and they look quite differently, which sort of makes sense if you think about burst of diversity that is a consequence of exponential growth, that you should have these sort of trees that if you unroot them, they will look closely to star trees or star genealogies. So these tall legs are kind of distinct feature, or the feature that distinguishes, for example, exponential growth from constant population size. And I'm showing it just to emphasize that it seems like there's information in these shapes of these genealogies, loosely defined shape. By shape, I mean some properties of the genealogies that gives us hope that we can infer population size changes, perhaps, by studying the genealogies probabilistically. Questions?
It all sounds great. This is a great intro to the coalescent.
Not of an Eric, okay. All right, but I'll take a compliment as well. So how it works in practice is that you can think of it as a kind of a sequential approach, which is, as I mentioned, not optimal. But the way information kind of flows is from sequence data, you have some hope of inferring genealogies. And maybe if these estimates, genealogical estimates, are very noisy, maybe you don't want to really estimate one and carry forward. Maybe you want to integrate over them. But nonetheless, the kind of information goes from sequence to genealogies, and genealogies encode information about population dynamics. And so that's how we, or at least I'm intuitively understanding the kind of the statistical information flow here. You can write down a big Bayesian model, and that's what has been done and is working great in many applications. Is that you can imagine that if you condition on sequence data, and you would like to infer everything jointly, the genealogy, Q matrix, which encodes substitution rates, changes in effective population size, and perhaps some other nuisance parameters, which I'm lumping into vector theta. Then you can write down this posterior up to proportionality constant. The first term, the probability of D or density of D, is the standard Feldstein likelihood. You just need genealogy and Q, and you can estimate the probability of observing the sequence alignment that you're starting with. You can put some prior and substitution rates. The prior in genealogies becomes this coalescent, so it's a coalescent-induced prior, although you could substitute some other models here, birth-death, for example, model. But so in this talk, I'm going to be talking about coalescent prior. Then you need some prior on this population size as well, and the prior and the hyperparameters. So after you define all these terms, you can turn the Bayesian crank and get the posterior using, for example, Markov chain Monte Carlo MCMC. So I haven't specified lots of these terms, but before I start specifying at least some of them, I want to say that I will concentrate in this talk on this red box, where instead of conditioning on data, I'll assume that genealogy can be inferred from the data without too much error, which is a limitation, and we would like not to do that. But usually we'd like to develop new methods under this simplistic assumption, and if they work, then there's some hope that we can actually do it directly from sequence data. If we run into severe problems, even by conditioning a genealogy, then there's usually little hope that we can even do the full posterior. So here we will assume that genealogy has been inferred and fixed, and we are trying to infer effective population size effectively from a genealogy. So genealogy becomes a fake data set. So in practice, two things to point out. First of all, that we would like to deal with genealogies which have sampling of tips at different time points. That's important when you working with certain types of applications. Two classes of them, two large classes, are these viral sequences, when you try to look at sequence RNA viruses, mostly HIV, influenza. Or if you are dealing with ancient DNA type of studies where you have some fossils and you extract DNA and you can date them, and so in that case you also have a tip that has a date which is, or tips that have different dates, and those differences actually matter. So that's one point, that we're dealing with this what's called heterochronous sampling or serial sampling. Another point is that there are many ways to approach the effective deparameterization of the effective population size trajectory. Here we assume a very simple discretization on a discrete grid. So we just assume that we have some grid points, and we have the step function that approximates whatever continuous function really is evolving in truth in time. Notice this is different from most popular models, let's say Bayesian skyline, in the sense that this grid is regular, so the grid is totally independent of the Coalescent events. Sometimes these models assume that the grid points coincide with the Coalescent events, and that's for convenience. We don't assume that. So our grid, we can resize it, and it can be size 100 or 1,000. And then effectively what's happening now, you have we still call it non-parametric, but it's a highly parametric model. I have now, if my grid size is 100, I have 100 little heights in my step function, and I'm trying to estimate them in this framework. So in order to do that, I need to write down the likelihood. And remember, my G is my fake data in this case, so I'm treating it as a likelihood. And conditional on gamma, which encodes these little heights. So gamma, little gammas will be heights of my step function. You can write down the likelihood of the or density of the genealogy, and this just comes from sort of standard coalescent theory from Griffith and Tavares paper, and extension to heterogeneous sampling by Rodrigo and Felsenstein. So we know how to do that, and it's been known for a while, but it's good. We have the likelihood, we can compute it. Notice those integrals are really quite simple because it's easy to integrate step function. So any questions on this busy notation slide?
It's good.
Right. So let me just give you a graphical representation of this estimation. So remember again, G is we had some genealogy G, which we can decompose into two important summary statistics. One is the vector of coalescent events, and here in this plot they are denoted by these heat maps. So instead of plotting each point, we blurred them into this heat map. So these, the dark little squares means that there were lots of coalescent events in these regions, and also sampling events. So sampling events were the times at which sequences were obtained to reconstruct this genealogy. And so we will be. So I just want to spend a little bit of time on that figure because we will be looking at many figures like that. So we will put these heat maps of coalescent events and sampling events on the bottom typically. The true population trajectory here was this dotted line, so it looks like a periodically changing pattern of ups and downs. And we reconstructed this this effective population size n of t or n e of t using the this non-parametric method, the multiple way of doing it. So we condition the genealogy and we reconstruct these little gammas. Remember, those are heights in the step function, and tau, which is a little hyperparameter which controls the smoothness of how similar they are. So in practice, we put like a little sort of a Brownian motion type or autoregressive prior on these gammas, so they don't change too quickly. They are not allowed to change too quickly by the prior. So this tau controls how dissimilar we're allowing the neighboring step functions to be, heights in the step function to be. And so this kind of brownish looking solid line, that's the estimated trajectory. This is a posterior pointwise posterior median, and then the gray areas will be pointwise credible intervals which form this credible band. And the figure is readable. I didn't forget to define anything here. Time on the x-axis, effective population size on the y-axis.
Yep.
So we can do this in this particular implementation of this Bayesian non-parametric phylodynamic reconstruction. It turns out it falls into a class of models. If you condition a genealogy, if you have a genealogy, it falls into this class of models which can be for which posterior distribution, the marginal posterior distributions, can be very, very quickly approximated without MCMC. Although we do have MCMC that works on this as well, but we can do this type of estimation. We can do in seconds, in a couple of seconds. So it's easy to use. It's better to use that fast method if you want to do lots of simulations. But the question that I would like to pose in this talk is, what happens if you sample more when your incidence is high, or when your population size is high? So what happens if you sample more? And you can see that this example that I'm showing is constructed this way, that we have more samples when the true and estimated curves are sort of high, and fewer samples when we have low effective population size. So in spatial statistics, that's called preferential sampling. And it also has many different names in other fields. In medical statistics, people study it as well. But that's potentially a problem, we think, and that's why we want to turn our attention to this.
I mean, if you do want a question, I mean, it seems like maybe there's sort of a systematic offset of your sampling events and your coalescent events in your figure. I mean, that's slightly different than sampling more when incidence is high, right?
Yeah, I mean, one is econ sequence of the other. So the simulations were constructed in such a way that first we have the true trajectory, then we generated random sampled events somehow so that we have more samples when population size is high. And then after that, we simulated genealogy just straight from the coalescent. And so this is just a property. If you distribute your sampling events in this way, the coalescent events will be, because you will have lots of sequences, and then they will coalesce quickly, but then you will kind of run out of them. So they will be nothing to coalesce for a while. And then you'll add the sequence, you add through the sampling, you will add more sequences, and then they will coalesce again. So it's something that we don't really control directly, but it just happens. So I would say it's a property of the coalescent in this setup.
Cool.
So I would like to point out two papers that looked at not exactly the same question, but something very related. Stack et al. Paper looked at different ways you can sample the infectious disease sequences during epidemics or before and after epidemics as well, covering the kind of the time period of an epidemic. Just trying to understand, they were looking more of, at this problem from a point of view of design. How should we sample sequences in order to maybe to estimate those trajectories reliably? Should we sample just uniformly across time? Should we target certain regions, maybe epidemic increase, so when we have increase in our number of infections, or which in this case is a proxy for effective population size, in this application is a proxy for number of infections. And so they concluded that they tried lots of different things, but one conclusion, and they have a lot of interesting things to say, but in terms of what's relevant for us, they said, oh, it's actually kind of difficult to estimate epidemic declines. And I want to highlight that because we will see a similar or related point in the results that we have. And also an interesting paper that we actually initially completely overlooked, and we're grateful to one of the two anonymous reviewers. So this reviewer pointed out that paper to us. Volz and Frost looked at, actually the main point was this birth-death model, which is a competitor or an alternative to the Coalescent model, which also defines a density for genealogies. But in birth-death models, you have to put the sampling model in explicitly. You can kind of ignore it in Coalescent, as I will show in a second, but it's hard to do it in the birth-death model, or maybe even impossible. And so they were interested, what happens if you misspecify your sampling model, how robust is birth-death approach to the modeling specification of the sampling distribution of your sampled sequences. But also, while doing that, they actually constructed what they called an augmented coalescent model, also which includes sampling, like the sampling density, which is almost exactly what we're going to do with the main difference is that they looked only at parametric exponential growth type of scenario, and we're going to look at it from a non-parametric point of view. And also, Volz and Frost didn't look at really effect of the, they were not interested, let's put it this way, in looking at the effect of preferential sampling on the estimation under the coalescent as much. They were kind of more focused on this birth-death model. So let me start defining these, what Volz and Frost would call augmented coalescent likelihood, and we're calling it this Bayesian on parametric phylodynamics with preferential sampling. So I like to view coalescent as non-homogeneous, inhomogeneous, continuous time Markov chain, kind of a death process, that every time you start with a certain number of samples and then you coalesce, so you kill one of the number of lineages goes down. So it's a kind of a death, Markov death process with sort of quadratic rates, which rates are quadratically dependent on the number of lineages at a time. And so that's where the same equation I showed before, so which defines the density of this genealogy. So that's one point process, this point where points are coalescent events. So it turns out only coalescent events matter for the purposes of reconstruction effective population size. It doesn't matter. You can scrap the genealogy and just keep the vector of Coalescent times, and you should be okay. If you keep also track of number of lineages at each time. We will model sampling events in time, right? So how we sample sequences as a inhomogeneous Poisson process with rate will be proportional to the power of the effective population size. So basically we want to mimic this behavior. Maybe you sample more when you have high population size and less if you have lower population size. One natural model would be to put just beta 1 is equal to 1. In this case, you would sample proportionally. So that's what we call proportional sampling. But we don't want to this beta 1 to be always 1. We just want it to make it like a fudge factor. Maybe it's not 1, maybe it's something else. We would like to estimate that. We will discretize everything anyway, so even though on the Poisson model you're not allowed to have identical time points, it violates one of the axioms of the construction of the Poisson process. But because we will discretize everything, it's not a problem for us. So once you have that, to the second piece, the sampling likelihood, you can now form the posterior distribution. But if you look more carefully, that's what we always do, or should do. We have two pieces of information, genealogy, but sampling times, even though they are part of the genealogy, we should not ignore them. So we're trying to say here, G or Coalescent times, it's one piece of information. Sampling times may be quite a second piece of information. Let's explicitly put S in our conditioning, in which case we can, using some reasonable assumptions, we can write down that posterior as proportional to the Coalescent likelihoods that we have seen before. And now the sampling likelihoods. But notice now in order to compute the Coalescent likelihood and sampling likelihood, I need to know my effective population size, this gamma, these heights of the step function that parameterize my effective sample size trajectory. And beta is these two beta 0 and beta 1 parameters lumped in one vector. And then again, some sort of maybe some Gaussian process smoothing prior on these gammas and some priors and hyperparameters. Usually we'll try to make them not very informative. So that's a new, so kind of this is an augmented model. That's what Waltz and Frost would call augmented coalescent. We're calling it BNPRPS. Notice what happens if your sampling distribution, if your density of sampling times does not depend on gamma. So you sample without ever looking at what your effective population size is, then this term becomes a constant, this probability of S given or density of S given gamma and beta. And therefore, you can drop it from the posterior and nothing bad will happen. It's just a constant, so it will disappear. You can just renormalize everything, you're fine. In MCMC, you will never even see it because your metropolis hasten ratio will cancel this constant. But when the gamma explicitly is there, when density of S explicitly depends on your population size, you cannot mathematically drop it. And by dropping it nonetheless, you have a model misspecification. That's probably the most important point of this talk. So I would like to pause maybe for questions here.
Yeah, I mean, makes sense.
It makes sense. Okay. So I would like to point out that it's similar.
Oh, sorry, Vladimir. I should point out that I hope people feel free to ask questions, and if they're interested in asking questions, you just have to move your cursor to the left-hand side of the screen, click on the Q&A app, and then turn that on. You can type
I cannot do it in the full.
Well, I think that's because you can't ask yourself a question.
Oh, I see. Okay. Oh, okay. That's fair. So I would like to point out, maybe to extend this pause just for a couple of seconds, is that exactly the same situation, for example, occurs in regression setting, where we, even though we don't believe that covariates are deterministic numbers, but we still condition on them. So the joint distribution of y and x, when y is response and x covariates, is, you can decompose it into a density of y given x times the marginal of x. And we're just assuming that marginal of x is nuisance. It doesn't depend on our regression coefficients and anything we would like to infer, and therefore we can just drop it. So we do this type of conditioning all the time. And so what I'm trying to say is that right now, all the phylodynamic methods that use coalescent implicitly do this conditioning and drop out the sampling times out of the picture, making this assumption, even though it's never really stated, this assumption, but it's there. The assumption is that sampling times do not depend on the effective population size, the distribution of sampling times.
Yeah, actually, Ralph just asked actually exactly that question, I think, which is that if we pre-specify sampling times, then we're good to go.
Yes, if we pre-specify them, we're good to go. But yes, this only matters if you imagine doing it repeatedly and repeatedly drawing samples in such a way that this distribution depends on the effective sample size. Yes.
I mean, I guess that you can take any distribution you like, for instance. But let's see. Hold on. The form of this distribution means that we can't say something like we have some delay, some time delay, for instance, in our sampling, because it's. Can we?
No, you can. You can. I mean, our current model does not take that into account. But if your true sampling distribution has some delay, some lag, you will still have a problem by ignoring it, right? So that's what I'm saying. And as a matter of fact, I actually mostly I don't want to focus too much on this model. This model is pretty naive at just a starting point. Our sampling model, this Poisson, the inhomogeneous Poisson model, you could and you should extend it further to be a little more realistic. My point is that right now we don't do any of that at all, and maybe we should be worried a little bit. I'll show you that we should be worried, but we shouldn't probably be retracting any papers.
So before we leave this point, Peter asks, even if sampling depends on population size and we condition on the sampling times, aren't we still fine just missing out on some information?
Yeah, so that's interesting. So that's and then we'll see that. So yeah, the question is it do we see the effect only in the precision or accuracy or both? And so we will see that actually it's both. You actually incur bias, and that's something that's it's counterintuitive a little bit, but you will have bias, and you also will not use some information that's available to you, and therefore you will have higher variance. But the effect on variance is larger, I should say in advance.
Cool.
All right. So let me show you how it works. So this is just an example of the. It's on the left. It's the same example I showed you before. So that's just regular kind of one of the Bayesian non-parametrics methods, but all of them will do something very similar to what I'm showing. And this is on the right. It's the same data, the same simulated data set, but the sampling times now explicitly modeled using this inhomogeneous Poisson. And notice in this case, as I mentioned, sort of the most noticeable difference is the width of the credible intervals. They're much narrower, but in some cases, bias is also there. So, for example, if you look at the time zero or close to time zero of the regular method, you'll see it's not only high variance, but also it seemed like it was biased, perhaps. Whereas this in the once you took preferential sampling into account into your model, you fixed both the bias and the variance. But that's the only point where it's actually obvious that both bias and variance kind of play a role. In all other parts of the trajectory, it's mostly the variance of the estimation that has decreased.
Is there a reason this bias only appears at the very last step?
Well, the reason is it's exaggerated because you have there is a reason, because there are very few coalescent events at this point, and because there's no sampling, there are very few sampling events at this point, so there's nothing to coalesce. So
Okay, but
coalesce,
if we had ended things on like an intense sampling event period, then perhaps we wouldn't see this so much.
yeah, exactly. So if you move time 0 to so it coincides with a peak, you will not see it. That's a very good point. As a matter of fact, we will separate later this initial point from the rest because the bias will be so huge here, so it will be hard to even see what happens inside the other parts of the trajectory. So we'll treat those two parts differently visually because they are on different scales. And again, this point is that if S of gamma and beta does not really actually depend on gamma functionally, then everything is good. Everything is good to go. You don't need to worry about this. Any questions on this kind of new way of looking at this preferential? So on the right is inference with preferential sampling being taken into account. So that's a we'll have more figures like that. So again, I just want to pause in case there are questions about sort of notation. All right. So just briefly, we want to do some simulation studies. So we coming back to Peter's point about we're going to do it repeatedly, and we will going to draw many genealogies, and every time we'll draw different sampling times. So that's important. Sampling times will not be pre-specified. They will be drawn randomly according to this Poisson process as we specified. So it will be according to our model. And then we're going to do two things. We're going to ignore this preferential sampling, or we're going to use it as part of the model. And we will look at sort of measures that measure bias and variance. And I'm probably going to point them out more explicitly on the figures. Probably will be better. And yeah, so in case of in terms of pre-specification, sorry, it's actually we could pre-specify them, but instead we drew them uniformly. So they're still random, but they're drawn uniformly across this interval. So this uniform distribution has nothing to do with the population effective population size, and that's kind of a null model of how we would like things to work. So let's look at this busy figure. So let me slowly kind of walk you through it. So on the top row, it's just the same figure we saw before, but without credible intervals, so it's not as busy. Again, sampling events are on the top heat map. So you see in the first plot, it's just uniform, so it's all kind of black with the same color. So everything is colored the same way. So it's uniform sampling, and these are coalescent events which are clustered because of this effective population size being periodic. And it's so basically there are three lines, but they're very close to each other. So again, the dotted line is the truth, and then the yellow and blue are the reconstruction methods. And in the case of uniform, they're exactly on top of each other. But if you move to the right, you'll see that when we start distributing sampling times proportionally to the effective population size, then the lines start to diverge a little bit, especially kind of near the valleys, near these regions, time intervals where effective population size is small. And you can see that they differ a little bit. So it's hard to see because of we wanted something realistic. So our effective population size kind of changes from let's say 10 to 100 or something close to that. So instead of staring at these differences in this plot, we just can move to the second row where we'll look at mean relative error. So this is at each time point we're measuring how off you are. 10% error, 20% error. So in this case, we see everything is sort of within 20% error more or less. In case of a uniform sampling, both methods perform identically, which is what we would like to see. And you have some error, some bias, which is fine. These are not unbiased estimators. They may be unbiased only asymptotically. And so we see some bias. That's fine. But if you look at this mean relative error plot, so I'm looking at the second row, second column now, you'll see that this bias goes up for the regular BNPR, which is just our vanilla coalescent model with non-parametric priors and everything in it. And then if you account for it, your bias goes down closer to the levels you see under uniform sampling. So it's maybe within 30%, but it's most of it actually within 20% error. Whereas the vanilla model, the regular coalescent, the bias can be as high as above 40%. Notice in time points that where this bias is where it's hard to see. It's where the population size goes down. So it's during the declines. And remember, I mentioned that this previous paper, Stack et al., also mentioned that the decline is kind of a problematic area. So we are in agreement with that paper saying that also for the preferential sampling, this period of decline, so if you're interested in decline and measuring maybe rate of decline of your epidemic or something like that, that's where you will incur the most bias if you don't account for preferential sampling. And on absolute values, it's hard to see. And my guess is that's why it even studies that have looked at preferential sampling before, formally or informally published or unpublished, probably kind of dismiss it because it's hard to see without looking at it relatively. So on absolute scale, it kind of looks very small. And also, just another point, the third row is this mean relative width of the credible intervals. So that there's something to do with precision, how precisely we can estimate things. And you can see that the methods agree, and your precision is about sort of 1, meaning that it's your width of your credible interval is about the same order size as your actual population size. So it's if your population size is 100, it will be sort of plus minus 50. That's your credible interval. It's actually more. So it's 1 only in some places. It's actually 1.5 or above in some other. And the precision goes down, or the variance goes up, or this width of the credible intervals goes up quite a bit if you don't account for preferential, if you have preferential sampling, but you don't account for it. But if you account for it, you kind of bring it back to the same level as it was in the uniform sampling. So is this first 2 columns? I just went through the first 2 columns. Are we okay with that?
Good for me. I'm curious about the the next step.
You're curious about what?
The third and fourth.
Oh, so the third. Okay, so third and fourth is this point. So notice we cut the time point is where, so that the here in this first 2 columns it ends. The time kind of ends if you look from left to right at the high point, and then we looked at this decline close to time 0 separately. So that's what it is. So the actual simulations run from 0 to whatever, and the first two columns end at 6. And so the third and fourth are between 6 and 0. And here, as I showed before, basically the coalescent has very hard time because there are not a lot of coalescent events there. So both methods have some bias because of that when you have uniform sampling, but under proportional sampling, the bias is exaggerated when you don't account for it, and you have huge variance. But you can bring both the bias down and the variance down if you account for preferential sampling. So it's exactly the same notation as on the left, but we're just looking. The first 2 columns look at time period, 40-something to 6, and then this third and fourth is 6 to 0. So just the effect is huge there, but I don't want to oversell too much this. Because it's kind of specific to this time 0 effect. So we wanted to separate the effect of being close to time 0 and lacking information versus everything else. Did it answer it?
Yep.
Okay, cool.
Probably soon we're going to see like something where it's not proportional or some sort of model in the specification sort of thing, right?
Yeah, so we have, I'm not going to show these results, but in the paper we do examine the effect of what happens if you misspecify this model. And yes, the results could be quite unpleasant if you assume some sort of more complicated preferential sampling, but you just use our simple Poisson, you will be off, and you also have bias. So,
is it worse than assuming that they're pre-specified?
It's a good point. I think I mean, not uniformly, but you could be better off sometimes by conditioning. I believe, yes, not accounting for it all sometimes could be better if you are totally off. If it's complete opposite, and the preferential sampling is you sample more when you have low values, and so which is complete opposite of what we're assuming here. I think you could be. Yeah, you can look at. I'll have the reference in the end of the talk, and I'll point out that there will be. We did this simulation as well. So I want to highlight this kind of counterintuitive point. It's a systematic bias could happen. Actually, it's interesting. One of the reviewers, the same reviewer who pointed out this paper by Boltz and Frost, also had a helpful comment. The reviewer said, "Well, I don't believe it. Maybe." You're seeing bias maybe because you're using, maybe the bias is a result of your, the strange Bayesian non-parametrics, your Laplace approximation. There's so many ways you can introduce bias into your estimation. So I want to say I don't think we're imagining it, although it's possible, but I don't think so. In order to convince that reviewer, we did a very simple parametric simulation with just exponential growth and decline. No Bayesian non-parametrics, simple MLE with 2 parameters, and we also see bias. It's also small, but it's there. So I don't think we are imagining it. So I'm going to show briefly 2 case studies. Both have to do with influenza, but one is focused on New York State, and the other across looks at more regions. So this is the same data set that Andrew Rambo analyzed in 2008 with the co-authors from the paper that I showed in the very beginning. And so we took just one genealogy from estimated from the sequence data. So we're conditioning on the genealogy, so it's a limitation. I want to emphasize this again. And so again, it's the same type of figures. We have BNPR, this regular, vanilla-based coalescent model, which conditions on sampling time on the left, and this sort of in this brownish credible interval solid line. And then the preferential sampling produced something which is kind of in the middle, which is the blue line. In the gray areas are credible intervals as before. And also sampling and coalescent heat maps are present as before. I mean, the main effect you can see, of course, you have shrinkage of credible intervals. That's what we expected. That's probably the most noticeable effect. But also notice that this BNPR, the regular coalescent, kind of fails to produce this periodic pattern. It kind of wants to produce periodic pattern, but sometimes it misses a year, or often that's what would be the case, or we just have lots of noise. This looks, I don't know which one actually is more accurate, right? I don't really know the truth in this case. I don't know the actual number of infections. But we think that this kind of cleaner periodic pattern seems a little more plausible. However, I want to point out that, remember this beta 1. This is the coefficient that we're using, that we are raising NE to the power of beta 1. So the most natural beta 1 value of beta 1 would be 1, right? Proportionally, you sample more. The more flu cases you have, the more you sample proportionally. We see this crazy number, which is about somewhere between 6 and 10, in this case, and we don't know if it's realistic, and we suspect this is just this beta 1 just acts as a fudge factor. The model is really not, the sampling model that we're using is still probably pretty naive. And so we absorb this model misspecification into this beta 1, but probably something else is going on there. Maybe if we included some covariates that we don't have, but should have, maybe this beta 1 would be closer to 1. And but right now we have only sort of one covariate in this case, this NE, the effective population size, that drives the sampling intensity.
Can you say a little bit about why the primary effect is in the confidence interval? It's not totally obvious to me.
Well, it actually kind of was also, even in simulation studies, the main effect was, it was not obvious, maybe the way we showed it, but yeah, the effect is credible intervals because you have the sampling times give you a lot of information about, especially with this high beta, that turns out the model wants the beta to be that high. It says, well, you have sampling times, therefore your effective population size, you have sampling events. Notice they're very clustered. If you look at the heat map, they are quite clustered. So I think that drives the.
Do you consider this to be cheating? No, I'm just kidding. But I mean, in a sense, it's funny. I mean, it's sort of like you could actually just like forget about the sequence data entirely and then make a predictor based on the sampling size.
That's true. I mean, one thing that just from sampling events, you actually can estimate only sampling intensity, but you cannot really estimate the sort of effective population size, which is still, which you can assume it's proportional something, but you wouldn't know any of these proportionality constants. So you can estimate from sampling times just the intensity, but then in order to go to effective population size scale, you would have to make a whole bunch of assumptions based on the data you don't have. So actually, you do need to, I believe still you need to combine the coalescent information and the sampling information, even when sampling information is kind of dominant.
Huh, okay, cool.
So this is less dominant case. I'm going to show you the, so this is looking at different regions. So USA, Canada, it's the same, it's kind of similar region that we looked at in the previous slide. But also we looked at multiple of them. I'm just showing 3 of them. The table on the next slide will have more of them. But you can kind of see a similar picture that, there's more periodicity inferred with the preferential sampling. But notice the sampling events are not as clearly clustered anymore. There's some clustering, but it's not as exaggerated as it was in New York State example on the previous slide. And as a matter of fact, if you look at South China, it's not even clear if there's clustering. It could be just sort of some noisy clustering of some sort. But again, we do have the again, shrinkage of the credible intervals. So Trevor came up with this another visualization. So we just kind of chopped each year and plotted each year on top of each other. And Mike Archer, who is the main driving force behind all that, calls this Trevorograms. I think that should be a thing. That's why I'm advertising it. So everyone should call it Trevorograms. And which is nice because you can kind of see periodic behavior being more consistent, right? So here, if you look at these mustardy lines, none of them, it's hard to see where they try to peak. Whereas it's more clear that they're trying to peak in winter each year in the USA and Canada, and the opposite is happening in Oceania, right? So it's
so these are samples from the posterior.
Those are just point estimates. So those are posterior medians, but they are the same curves on this plot. We just chopped each year and plotted each year on top of each other rather than next to each other.
Yeah, okay.
So instead of putting them next to each other, we put them on the top of each other. So it's so we can kind of see consistency of peakedness, let's say, of a curve, of all these curves across seasons. And South China has a peak in September, which turns out it actually does coincide with what other people think the flu season peak should be in South China. So it seems interesting and more consistent with epidemiology of influenza. And if you look at this beta 1 coefficients, they are not as crazy as they are for this Rambo data set that I described. They are somewhere between 2 and 5, or sometimes there's something between 4 and 8 in this Southeast Asia, but they're a little more tamed, but they're still above 1, which to be honest, going into these projects, I thought that we would be inferring something which is much closer to 1 when we looked at this beta 1 coefficient. So I'm still surprised, and I still think it's probably correct to assume that this beta 1 just absorbs some model misspecification of the sampling distribution. And this Poisson process distribution is a little too simple.
Or maybe it says that your sampling data is more powerful than your genetic data in some sense.
Yeah, but I think it's also probably fair to assume, for example, one simple model extension that we're working on right now is this beta 1. Maybe it's not realistic to assume that within each year, let's say if you think about influenza, that beta 1 should be the same constant across this year. Maybe you do sample proportionally in the winter when you have abundance of samples, but maybe you become more vigilant in the summer in the US and you sample not proportional anymore because you will see 0 samples there, but you sample, you exaggerate the sampling in the summers maybe. So I think it may have to do something with inhomogeneity, and beta 1 should be really a function of T maybe. But so that's the one conjecture that we have. So a couple of conclusions. I'm almost out of time. We think that unrecognized preferential sampling could bias you, but it's not going to be a huge bias, but you should be aware of it, especially if you are close to time 0 and you're following decline, right? So maybe you are trying to see the effect of some intervention and you assume that number of infections is going down and you're using these methods and you're close to time 0, you probably should be worried a little bit. Mostly, I think the promise is that you can improve your precision, but again, there's no free lunch, and you should specify your preferential sampling model correctly, which is probably hard, but we hope to increase complexity and realism of this model in the future. So maybe it will have more covariates that will account for some effects that have nothing to do with effective population size, but have something to do with maybe epidemiology or cost of sequencing, or what sequencing technology people have been using over the years and how that technology has been changing if you're looking at decades. We, of course, would like to stop conditioning the genealogy in this case because we think it seems promising enough to move it into BEAST. So that's on the agenda to put this preferential sampling model into BEAST. And also, we would like to develop some checks, probably in the context of these posterior predictive model checks. It's something that I think would be useful for preferential sampling. So you put this preferential sampling model in, for example, our simple Poisson model. You run your BEAST analysis, and then you do some posterior predictor check, and you look at it, and it's like, oh, my sampling model is awful. I shouldn't be using it. I shouldn't be trusting it. So we would like to do that. As a matter of fact, I think we already should be doing it with the Coalescent. We never really do any model check in the Coalescent in practice. At least I don't see a lot of it. And we probably should be doing it because Coalescent model is not God-given. It's quite possible that we are misspecifying it. It's probably most likely that we are misspecifying it all the time. So we would like to basically to be able to see two sides of this. Do this posterior model checks, and then kind of have some idea: is my Coalescent model an okay description of the data? Is my sampling model an okay description of the data? If yes, interpret it. If no, don't. And I will finish with some references. So this paper, the work I was describing is in this paper, which is on the archive right now, and was just accepted last week. It will appear soon in PLOS Computational Biology. And there are some important key references as well here. Thank you.
Very nice. I'm clapping.
Thank you.
Yeah, it's obviously a big step in the right direction for these sorts of epidemiological models.
Should we wait a little bit for questions from the audience, or there's no?
Yeah. So Peter says that he's having trouble with the Q&A app. According to me, it should be on. I guess people can also send Phyloseminar a tweet if people do that. Are there some other situations in which, like, are you also thinking about expanding this to other sorts of parameters of epidemics? I mean, I guess ancestral population size is the main one that people often estimate, but you could imagine something like maybe estimating parameters of an SIR model or something like that.
Yeah, so of course, instead of using, so right now we're using this Gaussian process prior and effective population size. We probably should be using SIR prior on this, and which will allow us to infer the infection rate and recovery rate and things like that. And people are working on it. I think I saw Eric Volz giving a talk on this recently. So, and their previous work that attempted that as well from David Rasmussen and Katja Coeli, I think. So yeah, so that problem will be there, but it will be, you can substitute a different prior model for effective population size. You'll kind of have a similar problem, and you will still have to account for preferential sampling. Of course, geographically, there's also, I think it has been known for a while that if you look at phylogeography, and if you oversampled some geographic areas, the phylogeography can give you very strange results as well. If you don't do it via coalescent with migration, if you do it just with the sort of kind of stochastic mapping type of approaches. And there's some workarounds on that, but it's a very related question. I think it's known to people who work on this in phylogeography, and maybe I don't really understand exactly how it's dealt with, but I know that it's there. It's a problem. People know about it, and somehow, kind of, sometimes at least able to get around it.
Well, if I understand Nicola's upcoming talk, it should. I mean, and if I understand what you're saying, I mean, it is going to be about phylogeography and have some stuff about bias sampling. So
yeah, hopefully, yes, it will be. Yeah, that's why it's a theme, right? But in this phylodynamics inference, right, that's also interesting, because even right now, if you look at Andrew Rambo's virological sequencing, that's exactly virological website where they looked at Zika data set. They have been reconstructing the sort of the migration events of Zika and trying to see where it came from to South America. But of course, sampling could be an issue if you oversample some areas or undersample some, and the sampling depends on some parameters you're trying to infer. So I don't know much about it, but I suspect you have to do something of the, to correct or to account for preferential sampling there as well.
Yeah. So Peter has another question, which is where are the samples from within each region? And I think he means geographic region here.
So you mean, I guess by that, if he means fine scale, I think they are all over the place from that region, but of course, probably with some geographical structure. But I don't know. I don't remember off the top of my head. So the details in this Zinder paper, PMC Evolutionary Biology, the last paper in that in my reference list. But I don't know exactly how geographically the samples are distributed within each region.
Yeah, I mean, I guess maybe this is also not something that you'll know about, but I mean, I guess he continues with a question asking, like, for instance, are they from the same like a given hospital, or?
No, I don't think they're just pulled from the database from databases, as I understand it. So I don't think they are from the same hospital. I think Trevor worked on this data set, so he would know more about this. But I think Trevor pulled these data from publicly available data sets like the flu database, and just classified them into different geographic regions post hoc. So they were not really. I don't think they're from the same hospital. That's what I'm saying. Mm-hmm. But geographical structure is an issue. I mean, all these models do assume, perfect mixing and fun mixing and everything, which is always suspicious to me when I think about where the samples came from and how much geography plays a role in the epidemics.
Yeah. So is there any sense in which this work would inform some sort of optimal sampling strategy?
I mean, yeah, I think so. Well, especially if you you could turn it around and ask a kind of a design question. And actually, this has been done before a little, not exactly in the same context, but there was a long work from Jeff Thorne and one of I think his former PhD students back in the day, when they looked at it from a design point of view, how you should distribute. They didn't look at effective population size. I think they wanted to reconstruct the rates, just substitution rates, and how best to distribute samples if you want to maximize your precision and things like that. So you could do it. I don't know if it would be doable just analytically. You could certainly do it with simulation, and that's actually part of what this paper that I mentioned, the Stack, Stack et al. Paper. That paper, protocols for sampling viral sequences. I mean, that's partly was their goal, to see where you could sample. But again, my point is that if you do that, you have to be careful, because maybe a protocol will say, oh, the most optimal protocol to do is that you should sample in such and such a way, and you will define it sampling in terms of your number of infections, or somehow in reference to epidemic time during the epidemic. And so I think you have to think about it a little bit and say, well, maybe I should now account for that in model. But I see where you're going. Yes, so it's definitely possible to merge it with a design question, how to design the sampling. We haven't done it yet.
Cool. Well, I think that's about it I haven't seen any questions come up on Twitter. So I think that we can just end here and remind people that the next talk is going to be from Nicola De Maio, University of Oxford, on Tuesday, March 1st. All right. Thank you very much, Vladimir.
Thank you, Eric. It was fun.